Okay, so let's go ahead and get started. Warn you that there's tomorrow, besides frog section and finishing up exercise 12, also 2 quizzes. So there will be a pre-lab quiz for exercise 12 and also a lecture quiz. Okay, lecture quiz for last week before, spring break. So I think I went up to here, is that right? Yeah. Okay, so it's going to include the synapsida, okay, that part of the animal phylogeny, that is the mammals, leading to the mammals, plus all of the tissues, plus digestion up to here, okay? So this is the material that will be covered for it, okay? All right, so let me just remind you, go back here to one thing that I want to emphasize, you might want to think about before tomorrow, is how to calculate amino acid utilization, okay? Because I will give you a problem, okay? I'm not going to give you all 20 amino acids, all right? It'll be restricted, usually when I do these types of problems, it's restricted to 4 or 5 amino acids, no more, okay? And so you might want to think about that. So I'm giving you fair warning. All right, so we are looking at the digestive process in mammals, and what we've done thus far is we've chased our food down to the small intestine, okay? And so I'm going to review this again. So we start in the mouth, the oral cavity, and already right there, besides mechanical digestion, that is fragmentation by chewing, we also have the first bit of enzymatic hydrolysis, and that affects only carbohydrates. So carbohydrate hydrolysis starts there with salivary amylase. Then it sort of discontinues as we get into the acidity of the stomach, because eventually that amylase is going to be destroyed because of the denatured. And furthermore, this is where we also have enzymes called pepsins. Okay, these pepsins are protein digesting, and amylase is protein. So it's going to get digested there too. So protein digestion starts in the stomach, but it can't be completed. Because one of the things about these proteases is that they don't cut every single peptide bond. Okay, they target a specific type of peptide bond, and therefore all you had was just pepsin to try to digest your proteins. You would just only have protein fragments, still too large to be absorbed into your body, okay? Because everything has to be broken down to the basic building blocks. For the carbohydrates, it has to be broken down to simple sugars, monosaccharides, and also the proteins have to be broken down to amino acids. Now, I'm sticking at this particular type of explanation because in reality, though, there can be taken into the cells and the epithelium of the mucosa tripeptides, maybe trisaccharides, in which they are taken in by endocytosis and then digested in a lysosome. Okay, there is a little bit of that, but most of the digestion happens in the lumen of the gut, and it is breaks everything down to the basic building blocks, and we're going to stick with that particular idea. Okay, because that's most of what we have is extracellular. Okay, first 25 centimeters in a human. Okay, happens to be the part of the small intestine that's called the duodenum, and this is a major place of enzymatic hydrolysis and also absorption. We'll get to that here in a moment. So this is also a place where there's secretions for the pancreas. So the pancreas is secreting all classes of hydrolytic enzymes. And I want you, all the enzymes that I've mentioned, I do want you to know what their general mode of action is and their source. Also, if it's a zymogen, what is a zymogen?
It's like the type of enzyme which like is inactive at first and you activate it later.
Exactly, and it has to be, so it's a protein that is inactive at first, and it has to be enzymatically cleaved, leads to a conformational change, changes the shape of the polypeptide, which then activates it. So I also want you to know what activates these zymogens. Okay, so the pancreas also secretes sodium bicarbonate, okay? Because the acidity in the stomach is a lot of times below it could be as low as 0.8 pH, really acidic. But by the time it gets into and released slowly, a little bit at a time, into the duodenum from the stomach, it's prevented, this acid chyme is prevented from going into the small intestine by the contractions of the pyloric sphincter. Okay, that's a circular muscle that keeps it tight, and eventually it'll open up and it'll let some of this acid chyme into the duodenum, and it needs to be neutralized. And so, because the enzymes here work at a pH of about 7, okay, so they would be denatured by this acid chyme. And that's what the bicarbonate buffer does, okay? Also, there's a horde of enzymes, or not enzymes, hormones here. Many of them, I'm not going to go into the details, I'll just mention one of them, but there's several different hormones that control the activity of the stomach and the duodenum, okay? One of these is a protein called secretin, so it's a hormone. What hormones happen to be is, because we'll go into hormones in greater detail later on, there are chemical substances that are released in one part of the body, and they have their target receptors somewhere else. And by the binding of the signal molecule onto a receptor, the receptor is always a protein. It's always a membrane, transmembrane protein. Transmembrane meaning it's in the plasma membrane, and it goes all the way across the membrane. So it has a part that works on the outside and a part that works on the inside of the cell. And by signal transduction, changes the behavior of the cell, leading it to change its behavior. In this case, secretin binds to cells in the pancreas and stimulates them to release hydrolytic enzymes as well as the sodium bicarbonate. All right? All right, now this is just a figure showing the relationship between stomach, liver, pancreas. Now, so our location of these things is somewhat similar to a shark, and you saw that yesterday. You looked for where the pancreas happened to be. From one shark to another, the size of the pancreas was really different. Just like people are different, sharks are different as well. And so you should have been able to find the pancreas in the fold, in the mesentery, just between the stomach and where it turns into the duodenum. So there's a bile duct from the gallbladder that also empties into a common duct from the pancreas that empties then into the duodenum. All right? All right, so what happens here is that, as I said, there's all the enzymes, it's in the duodenum, that we get complete enzymatic digestion of all 4 major macromolecules. Okay, carbohydrates, okay, we already started this in the mouth, but it was then interrupted by the stomach. But now there's another version of amylase. It's called pancreatic amylase. It is an isoform, okay? So when I say isoform, it has a slightly different amino acid sequence than the salivary pancreas, or amylase. And so both of these came from gene duplication, okay? So they come from separate genes, but they're very, very similar to each other, okay? And so this here will continue. Now the amylase, it hydrolyzes every other glycosidic linkage. And so it cannot break down anything like starch or glycogen into glucose. It can only digest it down to a disaccharide, maltose. Maltose consists of 2 residues of glucose linked together by a alpha 1,4 glycosidic linkage, right? But then there is another enzyme called maltase, and this finishes the job. Okay, maltase will hydrolyze maltose into 2 units of glucose. But we also eat in our diet, we also have, resulting from digestion, other types of disaccharides. Sucrose, lactose, okay? And to break these down requires yet other enzymes. Sucrase, okay, just to remind you, anything that ends with an OSE, and that suffix indicates that it is a carbohydrate. If it ends with an ASE, ase, that suffix indicates that it is an enzyme. Okay, so sucrase will hydrolyze sucrose into glucose and fructose, the two monosaccharides out of which it is composed. Lactase will break milk sugar, lactose, down into glucose and galactose. All right, so these are necessary. Now, in mammals, lactase is usually only produced until weaning. Okay, after a new baby starts eating solid food, then the production of lactase is usually cut off. All right. In humans, there's a little bit of an exception. There's a mutation, not in the gene itself, in European populations and also some East African populations in the world, in which it isn't the coding sequence of the gene, but it is in the primer or in the control regions that allow for lactase persistence, so that adults continue to produce this. All right. And it was highly selected for, especially in European populations and East African populations, simply because without this enzyme, the lactose in milk cannot be broken down, and therefore cannot be absorbed, and therefore it has absolutely no nutritional value for the human. It would go on down into the large intestine, where bacteria will break it down, and most of those bacteria are fermenters, and they produce a lot of gas, and so it's very uncomfortable. And also sometimes some of that gas is hydrogen sulfide, so it doesn't smell very good either. All right, so that lactase persistence allowed populations in the past that went through a lot of starvation conditions, that they could still utilize milk from the cattle. Cattle can eat grass, people can't eat grass. And so this is converting grass into cattle biomass, into milk that could be used. All right, so it was very strongly selected for. Now, these disaccharidases are produced by the epithelial cells of the mucosa, okay? And they're not released into the lumen. Unlike the pancreatic enzymes, they're in the lumen mixed with everything. Instead, they are attached, tethered to the apical plasma membrane. You need to change your notes because I noticed that I put this as extracellular matrix. It's not. The extracellular matrix is on the basal side of these cells. And so this is attached to the plasma membrane, okay? Plasma membrane that's facing the lumen. That is the apical side. Remember that epithelial cells display apical basal polarity. All right, so this is a little bit different. And this is makes sense because then these get these sugars, these disaccharides, they get hydrolyzed right at the surface where they are brought in, okay, to the cells. Okay, so now, so that's for the carbohydrates. All the carbohydrates will be completely digested in the monosaccharides here. But the pancreas will secrete many proteinases, and all of these are zymogens. Okay, they're secreted in an inactive form, and they have to be activated. So one of the most important ones here is the pair trypsinogen, trypsin. Okay, trypsinogen is the inactive form, and trypsin is the active form. Now, how does it get, now, pointing out, I didn't mention exactly where hydrolysis happens with pepsin. I just emphasize that it doesn't cut every peptide bond. Here I don't expect you to memorize this, you don't have to know it. I'm putting it up here only to emphasize that these enzymes don't cut every single peptide bond. There are conditions. Now in the case of trypsin, it will cut on the carboxyl side of the peptide bond when the upstream amino acid is arginine or lysine, okay? These are two amino acids that have a long side chain that is positively charged. Okay, so if it's a negatively charged amino acid, non-polar amino acid, or just a neutral amino acid, this enzyme will not cut that bond. Okay, so specific. There's another one here called chymotrypsinogen. When it is cleaved, or activated, it is chymotrypsin, and it cuts on the carboxyl sides of hydrophobic amino acids. Things like valine, glycine, alanine, all these have hydrophobic side chains. Okay? And so these two, just like with pepsin, would not lead to complete digestion of your proteins. It breaks them down into smaller fragments, that's all. Smaller fragments. Then there's another enzyme, procarboxypeptide. When it is activated, it is carboxypeptidase. And in the lumen, this one here, it does relieve, it can hydrolyze any sort of peptide bond as long as it is the terminal peptide bond on the carboxyl end of the polypeptide. So what do I mean when I say N-terminus, C-terminus of the polypeptide? What does that bring to your mind? Explain to me what is the amino end and the carboxyl end. Yeah, because these polypeptides, they're polar molecules, meaning they have a very specific direction. And one end terminates with an amino acid at the amino group end, and then the other one at the other end, the carboxyl group end, okay? And so this could do it. So if you had enough time, this would completely digest your protein. But it would take a long time if you had one of these others. Because by these other enzymes cutting a big protein into many smaller fragments, it increases the number of carboxyl ends that can be attacked, okay? Otherwise, if you had one huge polypeptide, it only has one carboxyl end, then it would take a long time to completely break it down. But if you break it into a bunch of smaller ones, you have many carboxyl ends that can then be digested. So this would do it by itself, but you would have to wait days before your meal is digested, okay? Okay, now, the mucosa. When I say the lining, I'm referring to the simple cuboidal epithelium that lines the lumen of the intestine. It also secretes some proteinases, one of which is aminopeptidase, and this is already activated when it is released. It doesn't have to be activated. And it, like carboxypeptidase, can completely break down a protein. However, it takes off one amino acid at a time from the amino end. Okay? All right. There's also produced, and this one here is tethered to the plasma membrane of the mucosal cells, dipeptidase. And there's several different types of dipeptidases. In fact, I think humans have about 20 different ones. They're produced and they're tethered to the plasma membrane. And what is a dipeptide? When you say a dipeptide, what's a dipeptide?
Peptide would be the amino acid excited to speak to.
Yes. Okay, it would be two amino acids covalently bonded together by a peptide linkage. Okay, so these still have to be broken down. And so these are attached to, as I said, the plasma membrane. And so we need all of these in order to have timely digestion of all the proteins. Okay, as I said, the aminopeptidase and carboxypeptidase, if that's all you had, it would work, but it would take you days to digest your protein. And you don't have days, okay, that really have to get it done. So all of them are real important here. All right, so what activates these, okay? There is another enzyme that is not a zymogen, okay? It goes by two different names, enteropeptidase. I prefer this, enterokinase. This is produced by the mucosal cells, releasing it into the lumen, okay, where it comes in contact with the inactivated forms of the proteins, of the enzymes. Now, this here is a kinase. Kinases are huge groups of enzymes. What they have in common is their catalytic activity. What do kinases do? Should be from Biology 101A. What are all kinases? What type of general reactions do they catalyze?
Oh, I was say like they do cell signaling.
They do what?
Cell signaling.
What about that?
Cell signaling.
Some of them can be involved in that, but I'm looking for something more general. Okay, not function, but the catalytic action.
Protein.
What?
Protein.
Well, it is a protein, yes, but I'm asking for its action. What type of reactions does it catalyze? When I'm talking about what type of reaction, hydrolysis reaction is a type of general mechanism. But what do kinases do? They don't hydrolyze. I even have it on the slide.
Phosphorylates.
They phosphorylate. What does phosphorylation mean? They use ATP as a source of phosphate, and they catalyze the transfer of a phosphate group to a very specific side chain on the protein. Okay, that's phosphorylation, transfer of a phosphate. Anytime you make any sort of change to a protein, adding something to it or taking it away, it leads to conformational changes, or it can lead to a conformational change. So in this case, the target is trypsinogen, and the heterokinase will attach a phosphate to a very specific amino acid side chain. That leads to a conformational change, and our trypsin is active. So this is how trypsin is initially activated. Then trypsin can activate all the other zymogens that I mentioned. In this case, it's by hydrolysis of a peptide bond. It'll cleave a peptide bond on pro-carboxypeptidase, converting it into carboxypeptidase, and so forth. And it will target all of them, even trypsinogen. Okay? So notice here that there's two different ways in which trypsinogen can be converted into trypsin. Initially, by phosphorylation from heterokinase. Secondly, others trypsinogens can be activated by trypsin cleaving a bond. Okay? Both of those lead to a conformational change, the same conformational change that leads to an activated trypsin. Okay? Two different ways. All right, so that's the proteins. They get completely digested down here. And the duodenum. Now, nucleic acids from the pancreas. There are nucleases, which will hydrolyze long chain DNA, RNA, into nucleotides. And there's other enzymes that will even break the nucleotides down further into nucleosides, and so forth. We won't go into too much more detail here. So here's a figure from a textbook, sort of summarizes the action of pancreatic zymogens and their activation. Okay, the sources that came from pancreas. Okay, so lipids. Okay, there's also pancreatic lipase that is produced by the pancreas, secreted into duodenum. And this here, what is a triacylglycerol or triglyceride? What is the chemical composition of triglyceride? They're also called triacylglycerides.
I know it's like 3 fatty acids, and I've got the top part.
Glycerol. So the top part is glycerol. It's a 3-carbon sugar. Okay, and to each of the hydroxyl groups that are on the 3 carbons, you hook a fatty acid chain. Okay, so that's a triacylglycerol. And so these, what type of linkage? What type of chemical linkage links the fatty acids to the glycerol? What is that linkage called?
Is it ester?
That's right. Ester linkage. Okay, so this here is what's hydrolyzed, and therefore you get glycerol and fatty acids, right? And there's other enzymes, lipases, that will also take care of phospholipids. Now, what does a phospholipid look like? Phospholipids are important because they're components of membranes. What would a phospholipid look like?
Phosphate and fatty acid, possibly fatty acid.
How many fatty acids?
2.
Two, yeah. So if you take a triacylglycerol and remove one of the fatty acids, attach a phosphate, and to the phosphate another polar molecule, okay, then you have a phospholipid. And so there are also lipases that are specialized for that, all right? Okay, now one of the problems is with lipids is that they're hydrophobic, largely hydrophobic, okay? And therefore, the medium in the small intestine is aqueous. It's a water medium, and therefore lipids are repelled by water, and therefore they will turn into large clumps of lipid, okay, droplets. Now, these enzymes can only hydrolyze what they can come into contact on the surface of that lipid droplet. And so one of the ways to break this down is from the gallbladder bile salts. Okay, these bile salts happen to be amphipathic, it means that they have a polar end and a non-polar end. These are detergents. They're just like detergents in many ways, because that's exactly what detergents are. They're amphipathic. They have a polar end and a non-polar end, and they can emulsify these large droplets into smaller droplets, okay, which increases their surface area. Even if we have a big lipid droplet like this, and we break it, don't change the mass. We're not changing the mass of it, but by emulsifying it into hundreds of smaller droplets, the collective surface area here is much, much higher than over here. And therefore there's more space here, surface area, for these lipases to work, okay? In fact, the fats are the thing that slows down your digestion the most. It takes the longest amount of time to break it down. Okay, all right, so now this fat hydrolysis only happens in the duodenum. All right, so what we've seen with carbohydrates is that there's two different places where their hydrolysis happens, in the mouth and also in the small intestine. For proteins, stomach and small intestine, but here for fats, only here. Yes?
For nucleic acids, since you didn't say anything before, does it also only happen?
It is, for the most part, it's only in the duodenum. But I do think that there's some, one of the things is that the nucleic acids do not take up a great volume of anything, so a lot of times we don't pay too much attention to them. But there is some enzymes in the stomach that do hydrolyze it, but most of it happens here. Okay. So here's a table that you might want to be familiar with. It shows the various sources of these enzymes, and for the carbohydrates, proteins, fats, nucleic acids, and I have pretty much discussed all these, except for I didn't go into the details of these that break down nucleotides. So you don't, if I didn't mention it, I didn't mention phosphatases or nucleosidases, and therefore you don't need to know them. But everything else here, I've pretty well covered everything in that particular figure. Okay, now after all this has been digested, it needs to be absorbed. As long as it's in the lumen of your digestive tract, technically speaking, it is not in your body. It isn't in your body until it crosses the mucosa, which is one cell layer thick. One cell layer thick, and gets into your blood, okay? Because right underneath the mucosa, the epithelial cells, which is a columnar, simple columnar epithelium, is a capillary bed. Any exchanges between body fluids and blood only happen in capillaries, not through veins, not through arteries, only capillaries. Capillaries are one cell layer thick, okay? So here we have the epithelial cells. This is the lumen. And of course, these are columnar cells, tight junctions, preventing anything from going in between. On the basal side, this is the basal lamina. We have interstitial fluid. Okay, so all your cells in your body are bathed in a liquid environment. Okay, and this is called interstitial fluid. It's also called extracellular fluid, but I prefer this. But right here, we have a capillary. Capillary, unlike veins or arteries, are one cell layer thick. And it is a leaky, simple, squamous epithelium. Cells are very flat, and unlike most epithelia, the tight junctions are not so tight. And therefore, what has to happen here is for any food, any nutrient has to be taken up for absorption to happen, has to be taken up into the epithelial cells, traverse across the cytoplasm, exit into interstitial fluid on the basal side, and then from the interstitial fluid into the bloodstream, into the capillary where it will be carried away. So this requires passing through, for any nutrient, passing through two epithelial layers, the mucosal layer and the capillary. Two epithelia, 4 plasma membranes. Has to cross 4 plasma membranes. This is going to be a physiological theme that we'll see not only here in digestion, but in respiration, in your kidneys, and in a lot of other places where things exchange membranes require the passage of something across 4 plasma membranes. Okay, where's the 4 plasma membranes? The apical membrane of the epithelial cell, mucosal cells, the basal plasma membrane, that's still the same cell, but still we have to cross a phospholipid bilayer twice into the interstitial fluid, and then across the capillary cell, the basal side, into the cytoplasm, then out the apical side, into the blood. Notice here that these two that are in contact with, these 2 membranes that are in contact with interstitial fluid are the basal side. For the bloodstream, the apical side of these cells faces the lumen of the capillary. Okay? All right, so now, another physiological property here is the rate at which things can be transferred from one medium to another is a function of surface area. Okay? Just like if you have a big auditorium for a rock concert with one little tiny door like this, it's going to take a long time to fill up the auditorium because there's a limited way of getting in here. But if you have a lot of doors, then more people can go through. It's the same principle here. So surface area is real important for exchange membranes, okay? So we have to have in our duodenum and also in other parts of the small intestine a large surface area to achieve the absorption in a timely fashion of the nutrients you're taking in. So this lining of the small intestine, if you take its collective surface area, it covers about 300 square meters. That's not square feet, that's square meters, okay? Meter is a little over 3 feet, all right? So it's about the size of a tennis court, all there in your small intestine. So how does it have that much surface area? for a tennis court to be in your guts, all right? And I'm not talking about the large intestine, I'm talking about the small intestine. So it prevents, shows a large surface area for this absorption. For one thing, this pipe is not smooth. It has large folds in it, okay? By those large folds, instead of being nice and smooth like that, it's thrown into large folds like that, okay? So if it were a smooth circle, the surface area is less than this curved one, all right? This creates greater surface area, but still, that's not enough. Each one of these folds are covered with finger-like projections of the epithelium that are called villi, okay? So here, this is showing the fold. This is a big fold. And then these little finger-like projections right here, there's thousands, millions of them. These are a villus. Villus is singular, villi is plural. Then, in addition, so this is here showing, we're taking this small area right here, blowing it up over here, and this is a villus. So it's finger-like projections, and there's many of them, they're right next to each other. But, so this villus is surrounded by the mucosal lining. That is the epithelium, okay? It is the simple columnar epithelium. Inside of the villus is a capillary bed, okay? So capillary beds, they result from the branching of a artery. It comes in, branches smaller, 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 until you have a network of capillaries, okay? And that is also for the blood system, increasing surface area. All right? Then, what's on the plasma membranes, the apical plasma membrane of those mucosal cells is that it is highly, highly folded. And notice here, my little diagram here, that I made the basal membrane just by one line, but here I've made a bunch of curves, okay, in the apical side. These are the microvilli. So the microvilli are folds in the apical plasma membrane, which further increases the surface area. So all these mechanisms together are for increasing the surface area for the purpose of food absorption. Okay, so inside the villus, capillary bed. Okay, so our whole task of absorption is to move these nutrients from the lumen of the gut across the mucosal epithelium into interstitial fluid, and from the interstitial fluid across the capillary epithelium into the bloodstream. Okay, so this is just another figure from your book. Large folds, upon which, if you take a little bit of this and look at it, you have a villus. These are villi. Okay, this is a fold, then a villi, and then when you look at a villus, just an isolated villus by itself, is that it has capillary bed on the inside, and around the outside is the epithelium. Okay, one cell layer thick, right? And this greatly increases the surface area. Epithelial cell, this is showing just one epithelial cell, but the plasma membrane highly densely folded, way more than what I had right here, okay? So that nutrients can go into the areas between these folds, but this here is the microvilli, right? Part of the apical plasma membrane of the mucosal cells. All right, so absorption. Three different mechanisms by which things can move from the, things don't just move by accident. They don't just, if all things are equal, things will pretty much stay put. And so to move something from the lumen into your bloodstream requires some sort of force, okay? And so one is that just simple diffusion. A simple diffusion requires a concentration gradient. All right, that means that on one side of the epithelium, the particle or nutrient has to have a high concentration, and on the other side, a low concentration, and things can diffuse down the concentration gradient. everything diffuses from high concentration to low concentration, all right? So if we have things broken down here to, we have a lot more nutrients here than over here, then we have a concentration gradient. Now, very few things actually enter. There are some that will enter across into the blood by simple diffusion, but most of the time that doesn't work. Okay, it's slow, okay? It's relatively slow, but most of these are sometimes moved by facilitated diffusion. So what's the difference between diffusion and facilitated diffusion in biological systems? Simple diffusion is from high concentration to low concentration. Incidentally, facilitated diffusion is the same thing, from high concentration to low concentration. But what makes it facilitated?
Using protein.
It has to go through a protein transport. Okay, the only thing that can diffuse from a high concentration to low concentration in your digestive systems are things that can cross through plasma membranes. That is, hydrophobic entities like short-chain fatty acids. Okay, they can go through, they're lipid soluble, so they can pass through there. But what about glucose? What's the problem with glucose? It's polar. It has a lot of hydroxyl groups, okay, and one carbonyl group. Those are polar. It won't pass through plasma membranes. It can pass through a protein transport, however. All right? And so some of them do. In fact, that's exactly how fructose gets in. It has a fructose transporter in the apical membrane that it can bind to, and the concentration gradient is the energy source to change the shape of the protein. All these transporters are carrier proteins, not channels. What's the difference between a carrier protein and a channel? What's the difference between a channel and a carrier?
Carrier protein uses ATP. It's kind of like that claw thing, right? One side goes to another.
Okay, but it doesn't necessarily have to have ATP.
Oh, yeah.
Okay, and what's a channel?
Channel protein is just, you have a channel to the plasma membrane ion transport.
Okay, so the shape of a channel protein, it has an aqueous pore. Okay, it can be gated. Okay, so there's part of the protein that can close the pore, and it leads to, and with certain signals, it can open the gate and allow things to pass through that pore down its concentration gradient. Okay, there's a lot of ion channels for sodium, potassium, chlorine. That's how they operate. Carrier proteins, however, have to change shape. They don't have a pore. So a ligand has to bind very specifically, shape, counter shape, and it will lead to a conformational change that carries the nutrient across. Now, if this is, now carriers can work both with facilitated diffusion and active transport, okay? Now, with active transport, it requires an energy source. For facilitated diffusion, it only requires a concentration gradient, all right? And it's always moving from high concentration to low concentration. Just binding of the nutrient to the binding site on the carrier protein can induce conformational change. Active transport can transport against a concentration gradient, but it can also transport with a concentration gradient, okay? And a lot of times, even though we have this concentration gradient, a nutrient's high here, low here, that a lot of them are actively transported anyway, all right? And the type of active transport in the digestive system is not direct primary active transport, but something called secondary active transport. Secondary active transport, I hope this is all review, is that, and carrier proteins only are involved in active transport, no channels. Whereas in facilitated diffusion, it could be a channel or a carrier. But for active transport, it's only a carrier. Now with co-transport, coupled transport, okay? It involves a minimally two different molecules. One of those molecules is the energy source. It has a concentration gradient, high in one side, low on the other. The other molecule is the molecule that gets transported, sometimes against its concentration gradient. And so this is driven by in the case of the intestine here, is by a sodium ion gradient. Okay, now there's a big important ATPase pump. Uses ATP. Okay, an ATPase is usually a protein that uses ATP as an energy source to do something. But what you normally find in cells of your body is that potassium is high concentration inside the cell, and sodium is high concentration outside of the cell. There's this disequilibrium. And what is the name of that particular ATPase brings that about? That this equilibrium between potassium and sodium. This also should be something you should have learned in 101A.
Is it just Na/ATPase?
Yeah, sodium potassium ATPase. It's also called sodium potassium pump. So it requires ATP energy to bring about conformational changes in which through one cycle, 3 sodium are pumped out of the cell, 2 potassium are pumped in. Okay, so that's what creates this sodium gradient. It's real important there. Okay, so this is something not in your textbook. Okay, this is something I put together showing just for glucose. So this is the epithelium, mucosal epithelium. Here's the lumen of the small intestine, cytoplasm, and over here, interstitial fluid, capillary is over here. I don't have it drawn in, but how do we get glucose into the cell? Because it's co-transported, active transport for glucose. Now, at the very beginning, so we have this apical membrane, and before all the starch gets digested, okay, at the very beginning, your glucose levels are low compared to the glucose levels in the cytoplasm. So do we have a favorable gradient here to facilitatively bring glucose into the cell? No, not at all, because the glucose levels are higher inside of the cell. It would be against the gradient. So we don't want to wait until all the starch is broken down so that the glucose levels is high enough on the outside. Glucose is very precious for living things. It's a major sugar you find everywhere. And therefore we want to catch as much of it as we can. So what we have in the apical membrane only, and I'm only showing one, but there'll be thousands of them, is a co-transporter called sodium glucose co-transporter 1. Okay, it's abbreviated sodium, let's see, glucose transporter 1. Okay, that's how it's abbreviated. I just want to write it all out there. Okay, so this is what we have in the apical membrane. In the basal membrane, we have our sodium potassium ATPase. Okay, now to create a sodium gradient. Now this is where these things are. This is only in the basal membrane, whereas this here is only in the apical membrane. Okay, apical basal polarity. The plasma membrane on one side, apical side, is functionally different than the basal membrane. They have different players. They have different proteins, all right? So let's assume that we have 3 compartments here. We have the interstitial fluid, the cytoplasm, and the lumen. 3 different compartments separated by phospholipid bilayers. All right, now if we start out with sodium concentration here in equilibrium. This is just an argument because we're not in equilibrium, but let's just assume it. Then once we put the sodium potassium ATPase into functioning here, it's going to pump out of the cytoplasm for every cycle, 3 sodium ions. Okay? What's that going to do to the sodium concentration across the basal membrane? Are we creating a gradient? So where is sodium concentration high? In the interstitial fluid. It's low in the cytoplasm. But what about across here? If we started out originally in equilibrium, we're pumping sodium out of this compartment. So now where is sodium high? Lumen, okay? So across the plasma membrane, apical plasma membrane, we now have concentration gradient. Okay, now I'm using that for just illustrating how we're getting a gradient. But remember, sodium bicarbonate is secreted by the pancreas. It contains sodium. So we have high sodium here. Sodium is the energy source for this co-transport. This here has a binding site for sodium. In fact, it has to bind 2 sodium ions and a binding site for glucose. When both of them will bind, then it leads to a conformational change. It's the carrier protein that drags glucose across against its gradient. Okay, so the energy source for co-transport, which is also called secondary active transport, is always a concentration gradient. And usually that concentration gradient is an ion gradient. So we have direct active transport here, where ATP is being used to create the gradient, and then the gradient is being used to bring the glucose in. Okay. Now, once we get the glucose in, we started with high glucose, it gets higher. Across the basal side, we can use facilitated transport instead of active transport. We have a high gradient here, it's low out here, and there is a carrier protein that called GLUT2 that has a binding site for glucose. Glucose will bind, and it's the concentration gradient that drives the conformational change without any further use of energy. This out here, glucose, now we have high glucose here compared to the blood that's coming in. And therefore there's a concentration gradient. We're not going to go into details here, but it's a facilitated transport. The glucose gets into the blood is moving and carrying the glucose away so that this gradient never builds up. All right, so this is how this glucose is getting in here. But what happens when your starch and glycogen that you ate finally gets completely hydrolyzed into glucose? Then we're going to have high concentrations here. All right, so we now have a favorable gradient. And so what happens here, the sensor for this gradient happens to be the sodium glucose transporter. It can tell when glucose is high versus low. It is a sensor, and it leads to a signaling process by which vesicles, transport vesicles that contain GLUT2 in the membrane, they were tethered just right under the plasma membrane. They will now fuse with the plasma membrane, inserting the GLUT2 transporter. That wasn't there to begin with. That wasn't there to begin with. Okay, so what am I saying here? Transport vesicles, tethered apical end, and they have the GLUT2 protein in their membranes. With the proper signal, these vesicles fuse and become part of the plasma membrane, inserting GLUT2. GLUT2 is a facilitated transporter, and therefore we have a favorable gradient now that glucose can just simply cross through this co-transport or through this facilitated carrier, and as glucose levels go up, and on the basal membrane we have GLUT2 It's always been there. It can just diffuse across down the concentration gradient. Once glucose concentrations get high. When glucose finally drops in the lumen, when glucose levels drop in the lumen, these are removed by endocytosis, okay? So that the GLUT1 or GLUT2 protein is moved. GLUT stands for glucose transport. Humans have about 12 or 15 different versions that are in different types of tissues, depending on it. But this is where GLUT2 happens to be, all right? Now, any questions on this? It's complicated, right? But that's life. That's just the way living things are. One thing I want to really emphasize here is that you should have learned in Biology 101A that plasma membranes are dynamic. They're constantly changing depending on the processes in the cell. This is a good example of where the plasma membrane is being remodeled according to circumstances, all right? Now, why not have just simply use ATP to transport these things across? It's because of the sodium gradient is an all-purpose energy source. Because how do vitamins get in? How do amino acids get transported here? They're all by transporters that run on, driven by a sodium gradient. These molecules all have different shapes. And so if you have vitamin B, one type of vitamin B, it has to have its own transporter, a sodium amino acid transporter, a sodium vitamin B transporter, and so forth. And they're all running from this concentration gradient. Okay? Yes?
So when glucose is high, why would we still have the GLUT2 facilitated transport when glucose is exiting the cytoplasm?
Exiting? Well, we still have to have a way to get from here to here. And it can't go through the membrane. It's polar.
Why would it still be facilitated transport?
Well, if the concentration is higher here than here, we have an appropriate gradient for it to go. Diffusion is from high concentration to low concentration.
Okay.
Okay, so we can go from high concentration to low concentration, but we can't go from what's facilitated from low concentration to high concentration. Now, even when we have this gradient, this here is still working, okay? And in fact, there's a lot of these transporters for various vitamins, various amino acids, that even though we have a higher gradient of those things here than inside of the cell, they're almost always being co-transported by active transport, regardless. And then once it's in the blood, all that blood carries it away so that the concentrations are always low in the interstitial fluids. Okay, now this is the way in which polar molecules are carbohydrates, amino acids, vitamins, that is water-soluble vitamins, are co-transported. This is a little bit different with lipid absorption because lipids, fatty acids are polar, or non-polar, excuse me, non-polar. They're lipid soluble. And therefore, they can, once we have broken them down into fatty acids, now what do you think a monoglyceride is? Talk about what a diglyceride happens to be, or triglyceride. What would a monoglyceride be? No, just scratching, okay?
One fatty acid.
Yeah, a glycerol with one fatty acid. Okay, now those are still lipid soluble enough that they can cross through the membrane. And so this is by just simple gradient, because our fatty acid concentration inside the cell is still low compared to what we have out here. And these could just pass right into the cell. Now they had to be broken down out here, indeed in the lumen. We had those lipases that remove the fatty acids from the glycerol. Glycerol, even though it is polar, it is also small. And therefore, being small enough, it can get across that membrane. Okay, once we get in here, then they are reconstituted inside of the cell. So this is a little bit different than for carbohydrates and the proteins and so forth. They're put back together as triacylglycerols, triglycerides, and then they're linked together, aggregated together with carrier proteins, okay? Because these lipids are not water soluble, all right? Furthermore, inside of these micelles, micelle is a collection of hydrophobic entities that form a droplet, okay, a solid sphere. But that is not water soluble, therefore they're attached to carrier proteins. The carrier proteins are polar, and therefore they make the structure, which is called chylomicron. It makes it soluble and able to carry in a liquid medium, okay? So you've heard of, when you've had cholesterol tests, maybe you guys aren't too worried about cholesterol yet, but there's high density lipoproteins, low density lipoproteins when they test you for cholesterol. That isn't just cholesterol. Those are cholesterol, fatty acids carried in carrier proteins, okay? So then, this is then by exocytosis at the basal side of the cell. This chylomicron is a lipoprotein, okay? This is the same thing. Lipoprotein is then exocytosed into the interstitial fluid, where instead of entering a capillary, these do not go into your bloodstream. They go somewhere else. They don't have, okay, let me see here. Let me go back. I'll go back to this in a moment. Let me go back here to another picture, get a feel on this. Villus. Okay, yeah. Go there. They cross into the interstitial fluid, but they do not enter the bloodstream. They enter another type of capillary called a lacteal. This is part of the lymph system. The lymph system is a parallel circulatory system. Okay? That we'll talk about a little bit later on. Okay, so there is a lymph capillary here, and this is where the lipids go. They go into that capillary, and then they are transported through the lymph system, not to the blood system. Okay, get down here. Okay, so these are attached to carrier proteins, and that includes cholesterol. Okay, it's not only the triacylglycerides, fatty acids. Now, the short chain fatty acids, they do get into the blood. Okay, so when I'm talking about short chain fatty acids, it's like propanol and acetic acid and so forth, butyrate, excuse me. About just only no more than 6 carbons. Okay, that's short chain. They are water soluble enough that they do cross through the membrane and into the bloodstream. But everything else, larger lipids, are carried by the lymph system. Okay? So even to get into this lacteal still requires crossing 2 plasmid membranes. The basal side of the lacteal, and then the apical membrane of the lacteal. Okay, now, I already asked that. This is going to be something you really need to get firm in your mind, because you're going to see this again with respiratory system. You're going to see it again with your kidneys. And you're going to see it again with your circulatory system. Is as things move between blood and other parts of the body, is that they cross through 2 layers of epithelium. 4 plasma membranes. 4 plasma membranes. Okay, now from your circulatory system, from these capillaries that are in the villus, they will join to form a blood vessel that is often called a vein. I don't like to call it a vein. It is called hepatic portal vessel. So I'm not calling it an artery, I'm not calling it a vein. What is distinct about portal vessels is that it is a blood vessel that each side of the vessel are capillary beds. Okay, arteries have the heart at one end and a capillary bed at the other. Arteries and veins both have the heart at one end and a capillary bed at the other end, and we'll get into that when we get into the circulatory system. The portal vessels are between two capillary beds. And I want to emphasize once again, capillaries are the only places where materials can move in and out of the blood, okay? Unless you're cut, all right? But under normal circumstances, that's the only place where things can exit or enter the bloodstream, all right? So this is a hepatic portal vessel, and where it goes is straight to the liver. This word, hepatic, means liver. And so the blood from your digestive system goes straight to the liver, and the liver regulates the nutrients, okay? Even after a meal, your blood glucose level, although it does go up right after a meal, it does not go up fantastically. That's because in the liver, that glucose is removed. So in the liver, there's a capillary bed, and therefore a lot of nutrients are then taken out and stored as glycogen, right? So, except for the lipids that I talked about, they go through the lacteal into a lymph vessel, and then they're dumped into the vena cava, a part of a vein right up in your shoulders. And the next stop, believe it or not, is the heart. So very bad design for modern high-fat diets, okay? Because it goes straight to the heart. Okay, all right, so homeostasis, what's homeostasis mean? Should be a concept you came across in Biology 101A. What is homeostasis? That's here in physiology, we're going to be talking a lot about homeostasis. What is homeostasis? How would you define it?
Maintaining internal.
Okay, maintaining physiological conditions within set bounds. Too high is bad, too low is bad. Things have to stay in certain bounds, and that's homeostasis. And it is dynamic homeostasis, meaning that it's always changing, but something, when things are going up, there's something that counterbalances to bring it back down. All right, so and that's true with these organic molecules. When they go to the liver, they're usually used, converted, stored. So a lot of the glucose ends up being taken out of the blood. And then from the hepatic, from the liver, then everything will exit the liver through the hepatic vein. These aren't the same thing. There's a hepatic portal vessel. And a hepatic vein, okay? Now, the hepatic portal vessel is going from your digestive system, the intestine, taking the blood to the liver. The hepatic vein is taking blood supply from the liver to the vena cava, which is another vein, important vein, into the heart. So this go to vena cava and into the heart. And we'll go into that in more details a little bit later on. Okay. All right, so all that small intestine, site of hydrolytic digestion and absorption. Large intestine, its function, okay, is to reabsorb water, because you have put all along that system a lot of water into, from saliva, into gastric juice, into everything, pancreatic juice. So about 7 liters. Your whole blood supply in your body is only about 5 liters, okay? So all this has to be gotten back, and therefore that is what happens in the large intestine. 90% of this water is reabsorbed, okay? Unless something goes wrong. So when the large intestine, there's an infection, it could be a virus infection, it could be a bacterial infection, it could be a food sensitivity, etc., allergy of some sort. If the food moves through too close, too fast, then that water is poured out and you have diarrhea. On the other hand, if something causes the large intestine to move too slowly, so that feces material is going too slow through the system, too much water is taken out and you have constipation, okay? So this is the function down here. Now, in mammals, where the large intestine, small intestine come together, there is an outpocketing, finger-like outpocketing, blind end sac, it's called a cecum, okay? And in humans and a lot of other, mostly predatory animals, in mammals, the cecum is very, very small. In humans, on top of it, it has something called the appendix. It used to be regarded as a vestigial organ that, when it is filled with bacteria, and sometimes it will become inflamed, and it used to be they just take it out because they didn't think it was important. But we now know that the appendix is not fully vestigial. It is part of, it has immune cells in it that contribute to your immunity. So, but in other animals, mostly herbivores, the cecum is really big because these things, just like in the cockroaches, those gastric cecae, blind end sacs, they contain symbiotic bacteria that can break down some of the food particles that you could not digest, okay? All right, then this is where, as water is removed, feces is formed, and this is all unabsorbed material, okay? The nutritional value of your food is only valuable, your food is only invaluable if you have the enzymes to break it down. That's one thing. Furthermore, that food is only valuable to you if not only that you have the enzymes to break it down, but that your intestine, small intestine, also has the transporters to get it into your blood. If you don't have the transporters for it, then it doesn't get into your blood. It goes on down to the large intestine. Okay, and this is where you have a lot of bacteria. A lot of these bacteria we're starting to understand are beneficial to your health, actually. They're really necessary. And so the feces is about 40% bacteria by mass. Okay. All right? Now, some of these will produce, as I said, some of them beneficial. They produce one vitamin, vitamin K. Now, this is interesting. The large intestine is not involved in absorption of food. Okay, that's a small intestine. However, vitamin K, being nonpolar, can cross through the phospholipid bilayers back into your blood. There's a concentration gradient. You have lower vitamin K. So we can absorb vitamin K in the large intestine. But anything else that if you have glucose, something wrong with your digestive system, and glucose made it down to your large intestine, you're not going to get it back. Okay? And it's just lost. Okay, so a lot of these bacteria are fermenters. And so depending on what's in your food, they're fermenting, they can produce gases. Sometimes that's just only methane. Sometimes it's carbon dioxide. It really depends on the type of bacteria. Sometimes it's hydrogen sulfide, which really makes the farts stink. Okay, so that's where all this is coming from. This gas is not stuff you swallow. Okay, it is produced by the bacteria in your large intestine. All right, so the very last part of the large intestine is the rectum. So things are stored there until stretch receptors in those muscles will indicate that it needs to be gotten rid of. Okay? Now, animals are heterotrophs, and of course, some herbivores, they're eating grass and plants that have a lot of cellulose, and no vertebrate can digest cellulose. No vertebrate can, in fact, most animals cannot. There's a few that can, but they don't have the enzymes to break this down. So how can, furthermore, things like cattle and sheep, they have a high demand for protein. What are the protein levels between animals and plants? Which one has more protein? Animal or plant? What do you think? What? Okay, which of these two groups of organisms, plants or animals, contain more protein? And therefore need more protein in their diet? Yes? Animals. Why? Why do animals require more protein? Why do animals have more protein? We require protein because we make protein, lots of, compared to plants. Why?
We're heterotrophic.
What?
We're heterotrophic, animals. Animals. are-
Well, herbivores are heterotrophic too.
We have muscle.
Muscle. We have a lot of muscle. Plants don't have muscle, okay? They need protein for making enzymes, of course, and structural proteins, but they don't have anywhere close to muscle like what we have. Okay, so how can a sheep or a cow eating plant material get enough protein to build their muscles? And in fact, where do they get the essential amino acids? Because they also have the same requirement for essential amino acids as we have. Now, it comes from fermentation chambers. Okay, in fact, there's two different mechanisms that herbivores do. Is they always have fermentation chambers and a load of symbiotic mutualistic bacteria, fungi, and protozoans. These have the enzymes to break down the cellulose and use it to make more bacteria, and the cow can digest the bacteria. Furthermore, the bacteria can make amino acids that the cow cannot. And furthermore, the bacteria produce a lot of short chain fatty acids, etc. And so there's two different mechanisms in vertebrates or in mammals. We just stick with mammals. Some of these are called foregut fermenters and versus hindgut fermenters. Foregut, hindgut fermenters. They have some sort of evolved some sort of chamber that's filled with bacteria, protozoans, and so forth that do the work for them. So the most famous one, so those that are foregut, usually these chambers are they evolved from the esophagus. They're alpalagines of the esophagus. Hindgut fermenters, they have a very, very large cecum. And this is where their fermentation chamber happens to be, okay? So if we compare like cecum is right at the junction between small intestine, large intestine. Herbivores don't have much need for a large cecum, whereas something that eats a lot of cellulose, they usually have a very large cecum. Okay, so this is one thing. Now, one of the problems with this, now, jump back here to foregut. Well, no, let's go back here. The disadvantages of being a hindgut fermenter is, yes, these bacteria, they will convert cellulose into amino acids, cellulose into other vitamins, perhaps even, but they will enter into the large intestine. Whereas the only site of absorption is really small intestine. This is a problem because, yes, vitamin K can go directly from across the wall of the small intestine, but not amino acids or any other products, and so they lose it. Now, a lot of these animals, such as rabbits, horses don't do this, but rabbits and some other things do, is that they eat their feces. Because now their feces are very nutritious, because it has all these amino acids that are produced by bacteria. Therefore, in fact, rabbits can produce two different types of fecal pellets. One which contains good stuff and they eat it. Others that it's waste and they leave it alone. Right? so this is what a lot of hindgut fermenters have to do. Okay, aren't you glad we're not hindgut fermenters? Okay, all right, politicians are. Okay, let's move on. Foregut fermenters. Okay, foregut fermenters. They usually have some sort of modification of the esophagus, and the most major group here happens to be ruminants. So this is an evolutionary clade within the mammals. It includes giraffes, sheep, goats, cattle. They're all ruminants. They've evolved several chambers, and this is a highly modified esophagus. And these are fermentation chambers. And I think that, yeah, this would be my last slide. So when they swallow food, it goes into a big chamber. It's called a rumen. And here it's contained with all sorts of bacteria and protozoans, as well as fungi, that break down the cellulose. And furthermore, they multiply, you know, because they're not doing this just for the cow, they're also doing it for their own metabolism and reproduction. So the amount of bacteria increases, okay, the numbers. These will regurgitate it and chew it again. So this is called chewing the cud. It's stomach or rumen contents, and they may do this several times to help break down the food because it's high in cellulose fiber. Eventually they'll move into something called a reticulum and do another pouch, and you don't have to know all these pouches, okay, and what happens there. But eventually they go into something called the omasum, which is the stomach. Produces hydrochloric acid, produces pepsin. And so what is being digested here is the bacteria, the protozoans. They can be digested, whereas the cellulose could not. So this is converting cellulose into microorganisms. Microorganisms then can be digested. Furthermore, these microorganisms also produce lots of amino acids, including the 8 essential amino acids. Where's the nitrogen source coming from? Well, these ruminants have evolved a way that instead of getting rid of the ammonia that is produced from the deamination of used proteins, amino acids, is that it's circulated into the blood and dumped back into here. So that to make amino acids, you have to have the nitrogen source. And so it's coming from the cow's own metabolism. Instead of losing it, that nitrogen in their urine, it is dumped here so that the bacteria can use it to make amino acids for the organism. Okay, so that's the digestive system. Any questions? I was hoping to get into the next lecture, but I'll have to leave it. So since it's so close to quitting time, it's no point in starting the next lecture. You get 5 minutes of freedom. Yes?
Can you explain one more time the difference between visibility and microvisibility?
Okay, this terminology gets a little confusing. What is the difference between villus, microvillus? Okay, so our stomach, or the intestine, has these large folds, and on these large folds are also finger-like out pockets. Those are the villi. That's a villus. One of those is a villus. So it is shaped like this, has it's a columnar epithelium, simple columnar epithelium, surrounded all around it, way around it. That whole thing is a villus. If we look at the cell, the epithelial cells that form that outer covering, the apical membrane is highly folded. This is the microvillus, is the apical plasma membrane of those cells. Does that make sense?
Yeah.
Because the two terms are not too close. Because the villus contains thousands of cells. One villus contains thousands of cells. But each cell has a plasma membrane that's highly folded on the apical side, which constitutes the microvillus. Okay? Right? Yes?
Can you go over the more plasma membrane that must be crossed again? You said four plasma membranes and 2 layers of epithelium.
Okay,
but
I'm
then-
too far away, I can't hear. Say that again?
Can you go over how like the 4 plasma membranes that must be crossed? Because you said 4 plasma
Okay.
membranes and 2
Why 4 plasma membranes? Okay, how come there's 4 plasma membranes? Okay, so here is an epithelial cell. Stuff has to go, first of all, across the apical plasma membrane. That's one membrane. It's now inside of the cell, cytoplasm, but it has to get out the basal side, which is another, it's a basal plasma membrane. And therefore now it's an interstitial fluid. To get into a capillary, it has to cross the plasma membrane, basal plasma membrane of a capillary cell. Now it's inside of a capillary cell. Then it has to pass out into the lumen of the blood by crossing the apical plasma membrane of that cell. So both of these are epithelial, okay? The simple columnar epithelia of the mucosal lining, and then the capillary cells are another epithelium. So there's 2 epithelia that have to be crossed, which means going into a cell and going out of the cell. To go into the cell and going out requires crossing phospholipid bilayer, a membrane, okay? So that's why there's 4 plasma membranes that have to be crossed. Okay, does that make sense? Okay? Yes?
On slide 36, I saw that like it was numbered a little bit differently.
36?
Like, yeah, slide 36, like the glucose high.
Oh, okay, let me go back to that. This one?
Yeah, why is like so 3 there, sure, is that supposed to be?
This 3?
Cause group 2.
Because I mentioned 1 protein, 2 proteins, 3 and 4. So these are just only numbers. I put them up there to say that we have these 4 special entities there. So this number in front really doesn't mean anything. Okay, it was just the way in which I presented these, because I first of all talked about that, then this, then that. So those, just ignore those. Okay, yes?
Can you go over, if you have enough time, that amino acid utilization?
Okay, let me go back, what slide is that?
Slide 8.
Okay, so let me go over this one more time. So let me just emphasize that the food that you eat may have an amino acid ratio, the ratios between amino acids that differ from your collected proteins. And therefore, for example, mushrooms are usually very high in a particular amino acid. I don't remember exactly what it is. It has a high amount of that amino acid that we don't need that much of it. And so if you consume a lot of it, then it doesn't fit in with the ratios that you have. So usually give this problem only in only 4 or 5 amino acids. I never go further than that. The real key here is the essential amino acids, because we can't make them by transamination. Okay? To make a non-essential amino acid, you still have to have amino acid to do it. Okay, so if you take in excesses of one amino acid you don't need, and you have too little of another amino acid that you do need, you can take the amine group from the excess amino acid and pass it over to appropriate sugar that will make the other amino acid that you're lacking. If they are non-essential, we have the amino transferases to do that, but we don't have the amino acid transferases to catalyze that type of reaction for 8 different amino acids. And therefore, we can't make it by transamination. Therefore, we want to maximize the amino acids, the essential amino acids, because they are the ones that are limiting. There are certain circumstances where it can be non-essential ones, but I'm always going to be giving you problems that involve essential amino acids as a limiting factor. So if this is the ratio you got in your food of those amino acids, 1 to 1 to 1 to 1, but you need 1 to 5 ratio between lysine and alanine, and 1 to 1 between lysine and glycine, a 1 to 4 ratio between lysine and valine. You can't use all the essential amino acids, but you need to maximize it. Okay? So the ratio here between lysine and valine needed is 1 to 4. You need 4 times as much as of this as lysine. So if you try to use all lysines, then how many for this, you have to incorporate, how many valines do you need? 4 times more. So if you try to use all these 20, you need 80 valines. You don't have 80 valines. You can't make them either. So this is the limiting factor right here. So first of all, compare your two, because I want to give you 2 essential amino acids, problems like this. Compare their ratios and figure out which one is limiting, okay? So we want to use all of the valine, okay? So if we use all of the valine, all 20 of them, then how much lysine do we need? 5, okay? So we need just only 5, back to the school over here. We just need 5, and we have 20, okay? So that means we can't use these 15, because we have to have a 1 to 4 ratio. Likewise, the ratio here between, once again, valine and glycine. If we use 20 of these, we need 4 times less for the glycine, and we have plenty of that. We have these left over. For the alanine, that's a little bit different here, because now we need for alanine, between valine and alanine, is we need 25, right? We don't have 25, okay? But this is a non-essential amino acid. We can use all the 25, but we have this left over and this left over, either of which could be used to take an amine group and transfer it onto the appropriate alpha keto acid to make the 5 other we have, okay? And so that's what we can use, and we have 25 that we can't use. So the calculation of the net amino acid utilization in the food is the ratio of what you used as a function of how much was in your food. We could only use, out of these 80, we could only use 55 because of the limitations of the essential amino acids. And that goes to 68.8% amino acid utilization. Okay? Yep.
So I don't know if you like clarified it, but if like the order was like switched around, like for the amino acids, would valine still be limiting, or like is that ratio?
It could be, okay. What tells which one of these are limiting is the ratio of one to the other. Okay, so in this case, you can't necessarily go by the numbers itself, except for here, you know, because if I had a lot of valine, 100, it won't be valine limiting anymore. It'll be the lysine. Okay, but you have to look at what do I have compared to what is required between them. And then first of all, ask yourself, okay, if I use all of these, how many lysines do I need? And do it the other way around. If I try to use all of these, how much valine do I have to have? And see which one works. Okay?
So when we go through this problem, basically the essential amino acids are what we first look at, since you want to make the most out of the essential.
You want to maximize how many of the essential amino acids that you